Welcome to the next episode of the Microbiology tube. So today we'll be talking about the serial dilution technique and its calculation. Many of the students are confused about the calculations of the serial dilution. So here I am going to make you clear about the serial dilutions and its calculations. So in the serial dilutions you will be taking a conical flask and a series of the test tubes. So what actually you do is that from the conical flask you will transfer the 1 ml of the volume of this sample to the first test tube, and from the first test tube we will transfer into the second test tube, and from the second test tube we will transfer to the third test tubes, and so on. So this is the procedure for the serial dilution. So what actually I have done here is that in the conical flask I have taken the 10 gram of the soil sample in the 10 ml of water. Actually, if you take the 10 ml of the water and if you put the 10 gram of the soil sample, so if you see the ratio, what actually happens in the ratio is that 1 ml of the water will carry the 1 gram of the soil sample. So here you can see, so this is the conical flask that consists of about the 10 gram of the soil sample in the 10 ml of the water. So during the transfer of the 1 ml of the water, what actually happens is that this 1 ml of the sample will carry the 1 gram of the soil. So here is this 1 gram of the soil. So 1 gram of the soil in the 10 ml of the 10 ml of the solvent, so or the 10 ml of the water, there is the 1 gram of the sample. So let's make you a little bit more clear from another slide, so which I have made it. So from the next slide you can see the dilution. So what actually I have done is that, so here I have put the I have taken the conical flask. So in that conical flask, I have put the 10 gram of the sample in the 10 ml of the water. So while transferring the 1 ml, you can see from here we transfer the 1 ml of the solvent. So you can see the transfer of 1 gram of the soil along with the 1 ml of the solvent. So whenever you are transferring the 1 ml of the solvent, so 1 gram of the soil is transferred. So what is the dilution for the first test tube? So what is the dilution? So for you can see here, so for the first test tube, you can see in the first test tube what is the dilution here. The dilution is 10 to the power minus 1. So how, from where this dilution calculation comes? So if you see here, so in the here you have in this test tube you have the 1 gram of the soil sample. So this is the 1 gram of the soil sample. So 9 ml initial volume was taken, and then 1 ml of the volume was transferred from the conical flask. So altogether there will be the 10 ml of the solvent. So what we do, we divide the total quantity of the soil by the total volume of the solvent, that is 10. So 1 by 10 means 10 to the power minus 1. So this is your dilution. So similarly for here, so from this test tube, if you transfer the 1 ml, so what actually happens, so how much quantity total is the 1 gram. So in the 10 gram of this test tube, so in this test tube there is the 10 ml of the water. In that 10 ml there is the 1 gram of the soil sample. So if you transfer the 1 ml of the soil sample from this one test tube to the 2 test tube, so to the next test tube, so what actually happens, so 1 that will be divided by the 10. So how much gram will we go? So how much grams? That will be the 0.1 gram of the soil sample will go. So from the first test tube, if you transfer 1 ml of the volume to the second test tube, then 0.1 gram of the soil sample will be transferred. Similarly, if you transfer the 1 ml of the sample or the solvent from second test tube to the third test tube, so 0.01 gram sample will be gram of the soil or the sample will transfer. So this goes on with the 10 to the power minus 2, 10 to the power minus 3, 10 to the power minus 4, 10 to the power minus 5 and 10 to the power minus 6. So this is our dilution. So again, so what actually you do? So after you make this dilution, so what you do, you will select some of the test tube in which you will do either the power plate or the spirit plate techniques. So in the power plate techniques, what we will do is that you will put the 1 ml of the sample in the petri plate, then after you will pour the petri plate with the molten agar. So then after you will solidify the agar and incubate, and after after the 24 hours of incubation, you can see the colonies. In the spirit plate technique, what you do, in the spirit plate techniques, you will distribute 0.1 ml of the volume of the solvent over the solidified media, and you will incubate it for 24 hours and you will count the number of the bacteria. So here you can follow either the power plate technique, you can follow either the power plate technique or spirit plate techniques. So after you do this power plate techniques or the spirit plate techniques, you will obtain the different types of the colonies, different numbers of the colonies and different types of the colonies. So after you do the serial dilution, so what you will do is that you will either follow the power plate techniques or you will follow the spirit plate techniques. So either you will follow the power plate techniques, this is the power plate techniques, or either you will follow the spirit plate techniques. So after following the power plate techniques or spirit plate techniques, you will count the number of the colonies. So here the number of the colonies from the first test tube is 150, and from the third test tube the number of the colonies is 35, and from the last or the sixth number of the test tube the number of colonies is 7. So you can see the decreasing number of the colonies. So decreasing number of the colonies signifies the decreasing number of the bacteria according to the dilution, because we have diluted the concentrations of the sample, and during the dilutions of the concentrations of the sample, the number of the bacteria will also be less. So after then, how will you count the number of colonies? So the number of organisms per ml, so that is what you have to calculate it. The number of organisms per ml means the number of colonies by inoculum size and the dilution. So I will make you clear about it. So the number of colonies means from the first test tube we have the 150 number of colonies, so that is the number of colonies. 0.1, here I have written the 0.1. so what is the significance? If you are doing the spirit plate technique, what we do, we spread the 0.1 ml of the sample over the solidified agar. So this is your inoculum size. So 0.1 ml is your inoculum size when you are following the spirit plate techniques, and dilution is 10 to the power minus 1, because we have taken the first test tube. Yes, you have taken the first test tube. You can see you have taken the first test tube, and in the first test tube, this is the first test tube where dilution is 10 to the power minus 1 and the number of colonies 150. So that is here. So that is what is here. So after calculation you will get the 15,000 CFU per gram. So if you do the spirit plate techniques, if you do the power plate technique, what you will do, you will distribute the 1 ml of the sample in the 1 ml of the sample in the petri plate, and over there the molten agar will be poured. So for that, so if you take the first test tube, so 150 is the number of the bacteria or the number of colonies. 1 is the inoculum size, because in the power plate technique you will put the 1 gram of the sample, but in case of the spirit plate techniques you will put the 0.1 ml of the sample, and 10 to the power minus 1. This 10 to the power minus 1 is the dilution, as I have already discussed, and from here you can see that you can get the 1500 CFU per gram of the colonies. So this is about the calculations of the spirit plate techniques and the power plate techniques. But you have to remember here, I have written here CFU. CFU stands for the colony forming unit. So what actually happens is that each bacteria is capable of forming one colonies. So one colonies means one bacteria is present, two colonies means two bacteria is present, and so on. And another thing is that I have written here gram but not the ml. Why? Because what is our sample? Our sample is soil. The soil sample is solid. So solids, these are measured in term of the gram, not in term of the ml. So we will write here the ml. So let's make you a little bit more clear. So as we are walking in the soil, so which can only be measured in term of the gram, so we have written, so the unit is CFU per gram. But if you are working in the solvent, suppose if you are finding the enumeration, or if you are finding the number of the microorganisms in the milk, so the milk are measured in the ml, not in the gram. So what you have to write? You have to write the CFU per ml. So in case of the solid, you have to write the CFU per gram. In case of the milk, you have to write CFU per ml in the milk. So this is the about the serial dilution techniques and its calculations. If you have understood, and if you like this video, please don't forget to subscribe my channel and don't forget to like this video. And if you have any confusions about this, please don't forget to comment. Thank you.